Dear students, in this session we will be learning about the genic balance theory, gynandromaps and Freemartin. I am Dr. Lata, Assistant Professor, Department of Zoology, Maharani Science College for Women, Bengaluru. So here when we talk about genic balance theory, it is something related with the affinity between these autosomes and the allosomes in order to determine the sex of an organism or to determine the gender of an organism, and which is well studied, taking Drosophila as an example. Now the concept of gynandromaps they are those entities which show the characters of both male and the female, and such kind of organisms are referred as gynandromaps. So we will learn in detail as to how these gynandromaps are formed and what could be their significance. And the other concept we'll be learning is Freemartin. So this is with respect to the calves, and we will learn like how these Freemartins are formed and what is their what result in the formation of Freemartin. Now here in the gynandromaps, if you can observe the structure of this butterfly, this is a male and this is a female, and if you observe the rest of them, they all share the characters of these two. It could be the wing pattern here, and here the mixed pattern of the wing, and here a little bit of this. So all these are showing the combination of both this male and female, and hence they are all referred as gynandromaps. Now the main objective of this session is to learn the concept of genic balance theory and to know how these gynandromaps and Freemartins are formed. And we will be learning mainly these three concepts here of genic balance theory, gynandromaps and Freemartin. Coming to the genic balance theory, it was first proposed by C.B. Bridges or Calvin B. Bridges in the year 1921. He studied this concept in Drosophila, wherein he suggested that the X chromosomes carry a factor for femaleness, whereas autosomes carry the genes for maleness. Now in Drosophila, there are 4 pairs of chromosomes. 3 are autosomes and the 4th one is an allosome or a sex chromosome. At first, the mechanism of sex determination was thought to be the usual XXXY type, and the males were heterogametic and the females were homogametic, meaning to say the males carry 2 different sex chromosomes, the X and Y, and the female carried only similar kind of sex chromosomes, that is they had 2 X chromosomes in them. Then when this non-disjunction of the X chromosome occurred, non-disjunction means both the sex chromosomes will not separate during the process of meiosis. So when such non-disjunction of the X chromosome occurred, the female produced resulted in 2 types of gametes or the ova, one with 2 X chromosomes because they fail to separate, and one without the X chromosome. So as a result, what happened? The offsprings that were obtained, they did not follow the expected ratio of 1:1. Rather, they gave rise to 4 types of different individuals, which were totally different from the normal one. Out of these 4 that were formed, one was a super female which had autosomes, 2 X chromosome and 1 Y chromosome, and one more was a super female having autosomes and 3 copies of X chromosome, and one was a male having autosomes and only 1 X chromosome. He was a male but sterile, and the other offspring was with autosomes and only one Y chromosome, which was non-viable. So, with this, Bridges said that the X chromosomes carry a factor for femaleness, whereas autosomes is the one which determines the maleness. And the major outcome of this was the Y chromosome does not take part in sex determination. So, C.P. Bridges, with this experiment, he came up with a very convincing explanation, and he explained it in terms of the ratio between the sex chromosome and the sex chromosome and the autosomes, correct. So, according to Bridges, the X chromosome in Drosophila carry this female determiners, whereas the autosomes, they carry this male determining factor in them. Okay, so keeping this as a background, he explained that when the ratio between the sex chromosome and the autosomes, the ratio between X by A, when it is equal to 1, it forms females, but if it is 0.5, then the offspring develops into male. Now, what if the ratio between the X and A is in between 1 and 5? 1 means female, 0.5 means male. What if the ratio comes in between? Then it develops into a intersex. It will be neither a male nor a female, but will develop into an intersex. And then what if the ratio is more than 1.1, like 1.2, 1.3, 1.5, then it develops into a super females. And when the ratio is lesser than 0.5, 0.5 means we said it develops into a male, and if it is less than 0.5, it gives rise to a meta male or a super female. So this table will give you a clear-cut idea as to what this ratio is all about and how the values are formed and what will it results in. See, for example, if you look here, the fourth and the fifth one, the number of X chromosomes are 3 and the autosomes are 3. So 3 by 3, the value is 1. So when the value is 1, it develops into a normal female. And here the ratio is 1, again it develops into a normal female. Now here if you see, the X chromosomes are 1 in number, autosomes are 2, then the ratio between X and A will be 0.5. When it is 0.5, it develops into a normal male. But then here if you see, the number of X chromosomes are 1, the autosomes are 3. 1 by 3, the value of this ratio X by A is 0.33, which is in between 1 and 0.5. So this develops into meta male or the super male. But here if you look at, the ratio value is 0.66, which is between this 1 and 0.5. Hence it develops into an intersex. Got it? Then here what happens? If you see, the value is 1, so it develops into a normal female. And here in these two examples, if you see, the value is more than 1. Okay, the value is more than 1, because of which it is 1.5, it develops into a super female, 1.3, it develops into a super female. So this is how C.B. Bridges, he concluded that the ratio between the sex chromosomes and the autosomes play an important role to determine the gender in case of Drosophila. I hope you got this concept right. Let's move on to the next topic, the gynandromorphs. Now, gynandromorphs are those forms which are neither male nor female, and they exhibit both characters of both male and the female. Now, if you look at this picture, you can see that you can identify a butterfly, a Drosophila, a lobster, and a fowl, and a man. But what is unique in this is when you observe it carefully, you can make out they show the characters of female and male in the same individual. So such forms which show the characters of both male and the female are called as gynandromorphs. Now let us understand what is the meaning of gynandromorphs. Gynandromorphs are the organisms which show both female and male characteristics. If you look at this term, gyne means women, aner means man, morph means form. So these are the forms which show both the characters of man and the women. Now where do you find these? These gynandromorphs, they occur in variety of species from butterflies to chickens to lobsters, and most notable in butterflies, moths and other insects. Now there are many kinds of gynandromorphs depending upon the position of sex tissue. The gynanders may be of the following types. They are of mainly three types. One is the bilateral gynanders. Here if you see, if you look at this picture, this is how the bilateral gynanders look like. You can see exactly one half of this will resemble a male or a female, and the other side with the opposite sex. So such kind of gynanders we call it as bilateral gynanders. Now this second type is anterior posterior gynanders. If you see, one sex tissues are confined to the anterior region and the posterior region showing the structures of the other sex. For example, if this is showing the male characters and this posterior region of the organism will show female characters. Such kind of gynandromorphs we call it as anterior posterior gynanders. And there's one more third set of gynanders which are called as sex piebald. Now here what happens, on the female body, this is a female on which you find patches of male tissue being present. Now this is a female on which you find the patches of male tissues here and there. Okay, such kind of gynanders we call it as piebald, sex piebalds. So these are the three main types of gynandromorphs. Like as we have already discussed, bilateral gynanders are the one which show the presence of both male and female characters in the same individual. So this picture will give you a clear-cut idea as to how these bilateral gynanders look in butterfly. Now anterior-posterior gynanders, we said anterior region represented by one sex and the posterior half by the other. And in sex piebalds, the body consists of female tissue having spots of male tissue scattered irregularly. You find such kind of thing in butterflies also, and in some birds like zebra finch also you find like this anterior-posterior gynanders are seen in zebra finch and also in few moths. Now what could be the reason for this gynanders? Now the main cause of this gynanders is they occur naturally, it is not an induced one. Naturally they occur, usually resulting from a random genetic error. Now when we say genetic error, what could be this genetic error? It is the non-disjunction of chromosomes during the process of meiosis. Then what happens, because of this the sex chromosomes will not separate properly, resulting in gynandromorphs. And the main significance of this gynandromorphs is that It helps in the studying of cell lineage and the developmental pathways during embryogenesis. And it is also said that because of this process of gynandromorphs, this gives us a clue to know why certain human diseases are gender specific. We must have heard like some diseases occur more in males or it is occur more in female, and why such phenomenon occur can be understood by this concept, by learning more about this concept of gynandromorphs. So I hope I made this clear to you all. Next concept that we have to learn is about free martins. Now, what's a free martin? In our initial slide, we said this is something related to a calf. Okay, so here they are related to calf, wherein when a twin is when twins are born, means to say when there is one male and the other calf is a female, then the male will develop into a normal male, the female will become sterile, and at the same time it also develops some male characters in it. So such kind of a female who is born sterile having male characters is what is called as a free martin. Now this free martin is an example of how hormones play an important role in determining the sex of an individual. Okay, so here why this female will become sterile, this is what is the main principle behind it. Now when these calves, when they are born, both male and the female, they are attached to the same placenta. Okay, they are attached to the same placenta, and here the male hormones are produced first, and once these male hormones are produced, that move from the male to the female through the placental blood vessel, because they share the common placenta. Because of the influence of these male hormones that are secreted first, and that when enters it into a female, then that particular female will become sterile. Okay, and because of the influence of this male hormones, it also develops a male characters, but it will have a normal female genotype but showing male characters. So such kind of a female is what is called as a free martin. But when if the twins are having both males, then absolutely no problem, both the males will be normal. If it is both females, then also it is fine. But when the twins are having one male and the other one to be a female, then this problem arises, giving rise to free martins. So this is the concept behind free martins. So because of this effect of male hormones on the female fetus, it results in partial development of male organs, and the female organs are incompletely developed. So even though the female calf is influenced by the male hormones only during embryonic period, its effect cannot be reversed. So hence it results in sterility. However, the male will remain normal. So that's how free martins are formed. So these are the textbooks that are that have been referred in order to put forth these concepts. And let's just take a few questions. Loss of X chromosome in a particular cell during its development results into option A, gynandromorphs, B, a diploid individual, C, a triploid individual, or both B and C. What's the right answer? It is option A, the gynandromorph. Question 2. The condition of free martin is observed in, is it seen in frogs, rabbits, cow, goat or sheep, or in man? You come across such condition in option C, that is cow, and rarely in goat and sheep also. Third one, in Drosophila, formation of gynandromorph and their cytological explanation provided best evidence that the X chromosome plays an important role in sex determination. X chromosome does not play any role in sex determination. Y chromosome is required for maleness, or Y chromosome does not play any role in sex determination. So the correct option would be answer C. Y chromosome does not play any role in sex determination. The fourth question, according to genic balance theory, X by A is equal to 1.5 will make what kind of individual? So if the ratio is 1.5, what does it lead to? Option A, male. Option B, super female or meta female. Option C, intersex. Option D, none of the above. And the right answer is option B, super female, because we learned that if the value ratio of this X by A is 1, it forms a female. If it is 0.5, male. If it is between 0.1 and 0.5, it is meta male. If it is more than 1, it is a super female. So since here it is 1.5, it gave rise to super female. Now the last one, the genic balance theory was proposed by Balbiani, Bridges, Fleming, or Mendel. Who proposed genic balance theory? It was proposed by C.B. Bridges. So the right option is option B. So I hope all these three concepts are easily, I've put it as simply as possible, and I hope you all understood this. Thank you for listening to me.